Hi viewers, welcome to SEKHAR'S Bio Factory for NEET. Principles of Inheritance and Variations with Chromosomal Theory of Inheritance. If you like this video, subscribe my channel and click on bell icon to get notifications. Like, follow and share my videos to your students and friends to get more information in biology related to NEET and other national state level competitive exams. In this video, I am going to explain the chromosomal theory of inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it is remained unrecognized till 1900 year. Because the main reasons for Mendel failure is the first reason, the communication was not easy in those days and his work could not be widely published. Secondly, his concept of genes like factor as a stable and discrete unit that controlled the expression of a trait. In similar way, the pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. The third reason is Mendel's approach for using mathematics to explain the biological phenomena was totally new and unacceptable to many of the biologists of his time. So Mendel's works suggested that the factor was a discrete unit. He could not provide any physical proof for the existence of factor or say what they are being made up of. So these are the reasons for Mendel failure. In 1900 year, the three scientists, De Vries, Correns and Steinmark independently rediscovered Mendel's result on the inheritance of characters. At this time, due to advancement in microscope that were taking place, scientists were able to careful observation of cell division. It leads to the discovery of a structure in the nucleus that appeared to double and divided just before each cell division. These are called chromosomes. In 1902 year, the chromosomal movement during meiosis had been worked out by two cytologists, Walter Sutton and Theodore Boveri, and also they noted that the behavior of chromosome was parallel to behavior of gene and used chromosome movement to explain Mendel's laws. The parallelism between gene like Mendelian factor and chromosome. Both the chromosomes as well as Mendelian factors are transmitted from one generation to next generation in an unaltered form. A trait is represented by only one Mendelian factor inside a gamete, especially a gamete similarly contains a single chromosome out of a pair of homologous chromosomes. The somatic cells are diploid chromosomes with a pair of chromosomes called homologous chromosome. So each cell having a pair of chromosome. In this pair, one is came from father, one is came from mother. Such chromosomal pair is called homologous chromosomes. When we observe the behavior of chromosome and gene, the chromosomes occur in pair. In similar way, genes also occur in pairs. Segregate at the time of gamete formation. In similar way, gene also segregate at the time of gamete formation and only one of each pair is transmitted into a gamete. The chromosomes independent pair segregates independently of each other. At the same time, on segregate the gene, one segregate independently of other pair. So it is a parallel behavior in between chromosome and gene observed by Sutton and Boveri. Walter Sutton observed that the separation of chromosomes into daughter cells during meiosis division. This observation it leads to the development of the chromosomal theory of inheritance. Meiosis and germ cell formation in cell with 4 chromosomes. Let us discuss now how the chromosomal separation or segregation takes place by taking 4 chromosomes in each cell. Before cell division, if the cell having homologous chromosomes called one maternal chromosome, one paternal chromosome, such condition is referred as a diploid condition. During interphase, the chromosomal replication take place. At this time, homologous chromosomes are replicated and each chromosome having bivalence. Each chromosomal pair having 2 chromatids. These are duplicated during interphase with the DNA replication. During meiosis 1, in first cell division, the homologous pair of replicated chromosomes are separated. So each daughter cell it receive only 1 chromosome. After meiosis 2 division, especially in second cell division, the sister chromatids are separated. Then in these 4 daughter chromosomes, the haploid chromosome number will be appeared. In this way, the chromosomes it will be duplicated and form chromatids. These chromosomes are separated in meiosis 1. At same time, the chromatid separates and finally it becomes into haploid nature in its nucleus. The independent assortment of chromosomes, especially during anaphase of meiosis 1, the 2 chromosomal pair can align at a metaphysics plate independent of each other. Let us discuss how the probability is there for the segregation of chromosomes, especially to understand the compare the chromosome of 2 different color in the left and right column. In these 2 columns, for example, in possibility 1, the chromosomal pair, one is represented with blue color, another one is represented with red color in similar way. The 2 equal probable arrangement of chromosomes at metaphase 1. So now in this possibility, the blue chromosome it will be present at the left and right side. It is the probability 2. During metaphase 2, during chromosomal segregation, so blue and red chromosomes are separated into 2 separate cells, but here alternatively one blue, one red, one red, one blue chromosomes are separated in possibility 2. During daughter cell formation, the first combination it may receive only blue chromosomes, second combination it receives only red color chromosome. It is a possibility 1. But in possibility 2, combination 3 having one blue, one red chromosome, possibility 4 having one blue and one red chromosomes. In this way, in both possibility 1 and possibility 2, chromosomal separation will be takes place. The important thing to remember that the chromosomes as well as genes occur in pair based on this observation. The 2 alleles of a gene pair are located on homologous chromosome site and it is referred as chromosomes. During anaphase of meiosis 1, the 2 chromosomal pair can align at a metaphasic plate independent of each other, and Sutton and Boveri they argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. That means due to having of allelic pair on homologous chromosomes, how the chromosomes will be separated. In similar way, alleles also can be separated in similar way, then it will enters into each gamete or pure gamete. The chromosomal theory, also known as chromosomal theory of inheritance or Sutton-Boveri theory, is the fundamental unifying theory of genetics which identifies the chromosomes as the carrier of genetic material. Sutton united the knowledge of chromosomal segregation with the Mendelian principle, and it is called as chromosomal theory of inheritance, which identified the chromosomes as a genetic material responsible for Mendelian inheritance. In this way, based on the observation of Sutton and Boveri, finally they concluded that the alleles are located on homologous chromosomes at a particular site, and how chromosome pair segregation takes place in similar way, allelic segregation also can take place during gamete formation. It is the main theme of chromosomal theory of inheritance proposed by Sutton and Boveri. Thank you for watching. If you like this video, subscribe my channel, like and share my videos to your students and friends.